Hello, I'm Tao Guo. I'm VP and Head of International Discovery Service Unit at WuXi AppTech. I have been with WuXi AppTech since 2008, and I'm based in Shanghai. Today, I'm going to talk about enable pharma and biotech innovation through open access platform. I'm going to focus on PROTAC drug discovery. My presentation today will include 3 parts. Introduction of PROTAC, WuXi PROTAC Platform, and future perspectives. First, let me introduce PROTAC. PROTAC is short for proteolysis targeting chimera. PROTAC is a dumbbell-like molecule. One end can bind to E3 ligase, such as VHL, CRBN, MDM2, or IAP. The other end can bind to a target protein with a linker between the 2 ends. PROTAC can bring the target protein and ubiquitination machinery into spatial proximity. As shown here, the PROTAC molecule first binds to E3 ligase with one end and the target protein with the other end to form a ternary complex. The E3 ligase catalyzes the ubiquitination of the target protein. The polyubiquitinated target protein then dissociates from the ternary complex. And finally, the polyubiquitin labeled target protein is degraded by proteasome to peptide fragments. PROTAC is catalytic in this process, and one PROTAC molecule can result in the degradation of many target proteins. The advantages of PROTACs are, it can degrade target proteins specifically with selective binders. It can target undruggable proteins. Its sub-stoichiometric activity requires low dose and concentration. And it can have prolonged duration of action. The first PROTAC paper was published in 2001 by Craig Cruz and Raymond Deshays. The first generation of PROTACs are peptide-based molecules that have low degradation and cell permeability. The second generation PROTACs are small molecules that have better drug likeness. In 2013, Craig Cruz founded Arvinas, the first biotech company focused on developing PROTAC drugs. Now, there are many companies working on PROTACs targeting a wide range of protein targets. PROTACs are heterobifunctional compounds with 2 recruiting ligands connected with a linker. A successful PROTAC drug should have the following attributes. First, it needs affinity and selectivity for the target protein to be degraded. Second, it needs good cell uptake, biodistribution, and suitable pharmacokinetics. Third, it should be optimizable, as the linker composition and the length are crucial variables in determining the potency. And finally, it should provide greater degradation selectivity with the binding selectivity inherited from the target ligand. Here are the key types of E3 ligands and the linkers for PROTAC that have been reported. There are 4 types of E3 ligands frequently used, including CRBN ligands at 59%, VHL ligands at 21%, IAP ligands at 15%, and MDM2 ligands at 5%. There are 4 types of linkers frequently used, including PEG linkers at 52%, alkyl linkers at 22%, rigid linkers at 16%, and cyclic and click chemistry linkers at 10%. PROTACs offer advantages over traditional small molecule inhibitors, antibodies, and oligos. For example, as compared to traditional small molecule inhibitors, PROTACs can target undruggable targets, can eliminate targeted proteins, and can be used catalytically. Given the advantages of PROTACs, many biotech companies have been founded focusing on PROTAC drug discovery, including, for example, Arvinas, C4, Chimera, Vividion, and others. Big Pharma have also started PROTAC drug discovery, either through collaboration with PROTAC startups such as Arvinas, or developing them in-house. Three PROTACs have entered clinical trials. Arvinas, androgen receptor degrader ARF-110, and estrogen receptor degrader ARF-471, and the BMS androgen receptor degrader CC94676 are in phase 1 clinical studies. Many PROTACs from Biotech and Pharma are advancing in preclinical studies. Now, let me introduce WuXi PROTAC Platform. First, let me give you a quick introduction of WuXi AppTec. WuXi AppTec provides a broad portfolio of R&D and manufacturing services that enable companies in the pharmaceutical, biotech, and medical device industries worldwide to advance discoveries and deliver groundbreaking treatments to patients. Our vision is every drug can be made and every disease can be treated through building the open access platform with the most comprehensive capabilities and technologies in the global healthcare industry. WuXi was founded in 2000 with only one chemistry lab, one customer, and 4 co-founders. Now we have a global platform with 29 sites worldwide. We have more than 3,900 customers and partners, and we have over 22,000 employees, including over 18,000 scientists. We have a global footprint with 29 sites across 8 countries and regions. Now, let me introduce Research Service Division, or RSD. RSD is an integrated discovery chemistry and biology platform led by Dr. Steve Young, our co-CEO. RSD has over 7,000 scientists in 6 units: IDSU, HIS, CAS, Bio, OIU, and HDB. IDSU provides synthetic chemistry and medicinal chemistry services to customers. HIS provides DEL and structural biology services. CAS provides analytical and purification services. Bio, OIU, and HDB provides in vitro and in vivo pharmacology services. RSD has built an integrated platform to enable PROTAC drug discovery. Our platform includes PROTAC synthesis and purification, pharmacological evaluation of PROTACs, and high-resolution crystallography of binary and ternary complexes. For PROTAC synthesis, we have established a modular platform to enable the efficient synthesis of PROTAC molecules. First, we have a collection of the commonly used E3 ligase ligand building blocks. For example, VHL, CRBN, MDM2, and IAP ligands. Second, we have a large collection of frequently used linkers in-house, including PEG linkers, alkyl linkers, click chemistry linkers, and rigid linkers. Third, we have also assembled a collection of over 100 linker plus E3 ligand precursors. Over the past 5 years, we have prepared over 40,000 PROTAC molecules for our customers. In addition, we have also scaled up kilograms of PROTAC candidate compounds for our customers. Our CAS team has a strong analytical and purification platform for PROTAC compounds. We have dedicated analytical and purification team with solid experience to support the purification and characterization of PROTAC compounds. We have a strong track record in purification. Over 40,000 PROTAC compounds using over 200 different conditions and a 95% success rate. For PrEP HPLC, we have experience in solving the PROTAC-specific purification problems, such as poor solubility and a poor stability issue. For LC mass analysis, we use UPLC and corresponding columns to identify impurity. And we have accumulated knowledge on the property and the stability of different kinds of PROTACs. For SFC analysis and purification, we use special columns for PROTACs, and we have developed novel purification methods for chiral PROTACs, purification with high recovery. RSD has a strong PROTAC pharmacology platform. We have implemented systematic support for PROTAC drug discovery through various in vitro assays and formats. Our biochemical and biophysics assay includes target PROTAC or E3 PROTAC binary binding assay, target PROTAC E3 ternary complex formation assay, and target ubiquitination assay. Our cellular assay includes target or E3 engagement assay, target PROTAC E3 ternary complex formation assay, target ubiquitination assay, and a target degradation assay. To evaluate E3 ligase PROTAC binary binding in live cells, we have nanobread method to measure PROTAC permeability and engagement with E3 ligase. As shown here, PROTACs that are cell permeable and engage VHL will compete with the tracer for binding to VHL, resulting in a decrease in bread signal. E3 PROTAC cellular engagement assay could serve as a powerful tool to evaluate the cell permeability and PROTAC engagement kinetics onto E3 ligase. Target PROTAC E3 ternary complex formation assay can serve as connection between binding and function. The alpha screen and alpha LISA assay is recommended for biochemical ternary complex formation due to relatively large complex formation involved. The key of ternary biochemical assay development includes selection of potent reference compound, usage of high quality protein, and identification of proper detection condition. The binary and the ternary assay is used for optimization towards preferred ternary complex formation. SPR assay provides rationale for preferred ternary complex formation to drive PROTAC SAR. SPR provides affinity and kinetics parameters to truly characterize binding events. Our BioCore 8K and BioCore 8K Plus are capable for both SAR study and library screening. The alpha value can be calculated as rationale for designing of PROTACs with preferred ternary complex formation. We have developed a luminescence-based HIBIT cellular system to evaluate target degradation. Our system is a two-in-one system, switching between endpoint detection and kinetic detection. It enables monitoring of target protein degradation at a physiological level and supports HTS, SAR, and PROTAC MOA study using the same cell line. We have developed, here shows our HIBIT cell line generation and validation. The plot illustrates the identification of positive 293 T cell line, large bit monoclone, the identification of positive target HIBIT knock-in monoclone, and the degradation of targeted protein with 4 reference compounds in the endpoint assay. We also have experienced scientists to support ADME PK and bioanalysis of PROTAC compounds. And we have evaluated over 1,000 PROTAC compounds. For ADME PK, we have tailored test systems for PROTACs, and we have accumulated knowledge and experienced staff to interpret ADME PK results. For bioanalysis, we have experienced in overcoming challenges associated with PROTACs such as poor solubility, high lipophilicity, non-specific binding, and poor stability issues. We have capabilities in protein production of E3 ligases and targeted proteins, and in crystallographic study of binary and ternary complexes. We have established a strong PROTAC platform at WuXi AppTec to support our customers' drug discovery programs. This platform includes chemistry, in vitro pharmacology, ADME PK, bioanalysis, and structural biology. And we can perform research from target degradation, ternary complex formation, mechanism, selectivity, to crystallography. Future perspectives. Despite rapid progress in PROTAC research, there are still challenges. Discovery of PROTACs remains empirical and little rationale guiding the pairing of specific E3 ligase with a given protein target. What percentage of a targeted protein should be degraded to trigger a phenotypic response? Acquired resistance to PROTAC treatment, degrade off targets that were untouched by their parent inhibitors. Despite these challenges, there are also opportunities. Preclinical and clinical studies demonstrated multiple routes of administration, including oral route, wide distribution of PROTACs to organs and tissues, including brain. There are direct binding assays for hit identification to develop E3 ligands. E3 DNA encoded library can be used to sample a vast chemical space. Fragment-based approach, screen a compound set from diverse and lower molecular weight pharmacophores. Structural-based design from initial hit to optimize the ligands. Molecular modeling, exploiting E3 ligands that are essential to the survival of cancer cells is a promising strategy to avoid resistance mechanism. There are over 600 E3 ligases, and currently only a few E3 ligases with ligands are used in PROTACs. So there are vast numbers of E3 ligases unexplored. It is desirable to extend the repertoire of ligands to E3 ligases with a variety of structural properties and diverse temporal and spatial expression profiles, essentiality in cancer, and ligandability. It is also desirable to be able to design PROTAC molecules using computer modeling. In addition to PROTAC, new degrader technologies such as LITAC, ALTAC, HITAC are emerging. These new degrader technologies can potentially cover a broader target spectrum. We are looking forward to seeing diverse modalities for targeted protein degradation. Thank you for your attention. Thanks.